So good afternoon, everybody. My name is Alberto Auricchio. I am the coordinator of the molecular therapy program at Patel Institute of Genetics and Medicine. And as a member of the board of the ESGCT, I'm here today to introduce you today's speaker on our e-seminar series on COVID-19. It's a pleasure to introduce you Dr. Patel, who is a assistant professor at Wistar Institute Vaccine and Immunotherapy Center, where Dr. Patel research is focused on development of vaccine and immunotherapy against emerging infectious diseases. And this includes the study of immune mechanisms that contribute to the protection. Dr. Patel research employs non-viral DNA vector for vaccine and antibody delivery. And her most recent studies and achievements include the development of DNA vaccine against the Ebola virus, MERS, and SARS, as well as several studies in which she has developed and employed DNA-encoded monoclonal antibodies. Her preclinical animal studies have led to the successful clinical translation of anti-Ebola DNA vaccine, as well as antibodies or DNA-encoded antibodies against the Zika virus. And together with the Wistar Vaccine and Immunotherapy Center, Dr. Patel is leading the preclinical immunological studies of a SARS-CoV2 DNA vaccine candidate, which has entered the clinic, and it took about 10 weeks from the vaccine design to FDA approval. And now the trial is ongoing. And so I welcome Amy, and I ask her to present her talk on this new technology for vaccine development, especially tackling SARS-CoV2 and COVID-19.
Great, thank you very much, and thank you for the invitation to speak during the ESGCT e-seminar series. So today I'll be discussing novel vaccine strategies targeting SARS-CoV-2. I focus mainly on DNA vaccines, but I'll give an overview first of some of the other many vaccine candidates that are currently being evaluated also against SARS-CoV-2 and COVID. And so I think there's been a lot of introduction during the e-seminar series, but I thought it would be a good idea just to go over some of the background. So back at the end of December 2019, a lot of reports started to surface about this new virus that was coming up in China. Nobody had much information about it. There were a few reports of different illnesses. The infectious disease community started to follow this and try and understand. This happens periodically, and we try to see whether or not this is truly an emerging virus, or whether it's something that's a one-off transmission between animals and humans, but doesn't have further human-to-human transmission. And so on the left-hand side, this was one of the first reports of this novel coronavirus with associated pneumonia in China. In terms of phylogeny, this new coronavirus is quite similar to SARS coronavirus, which there was an outbreak back in 2003, and that was transmitted between the civet cat and people, and it's also more distantly related to MERS coronavirus, which is another one that has been a very important coronavirus in recent years, especially in the Middle East. And so what we've seen in the past just a few months is rapid global spread around the world. So initially what started as an outbreak has now become a global pandemic, and pretty much every country around the world has been affected by SARS-CoV-2 infection and the associated disease, which is COVID-19. Many countries have had either local or national lockdowns to kind of restrict movement and enact social distancing to try and prevent further spread of the virus, and to minimize some of the symptoms that we're seeing as well. And so this happened quite rapidly in some places. Other places were able to enact some of these social distancing and lockdown measures a little bit earlier and did not have the great increase that other places have seen. And what we're still trying to learn a lot about SARS-CoV-2 infection. What we've seen so far is that it is a great spectrum of disease. So you can have everything ranging from asymptomatic infections and uncomplicated infections with mild respiratory illnesses, all the way up to severe illnesses. And many of the most severe cases result in acute respiratory distress syndrome and can be fatal. The time course of illness is also varied. So in the more mild cases, you have a very short time course to illness, while in some of the more prolonged cases, it can be greater than 10 days. And there's a whole range of clinical features as well, ranging from a mild cough to fever, fatigue, all the way up to respiratory distress. And in some cases, we've seen a whole range of new symptoms that we did not previously expect to be associated with COVID disease, such as loss of taste and smell as an example. And in some of the more severe cases, we're seeing lymphopenia as well as elevated D-dimers and coagulation markers, as well as other, not necessarily related to the coronavirus infection, but other host inflammatory mechanisms that are coming up in response to the disease. So in terms of now vaccination, of how we're going to approach trying to develop a vaccine, we kind of go back to the idea of herd immunity and vaccination. And so we need to find ways that we can train the immune system for protection. And so the idea behind herd immunity is that you have enough people in the population that are immune to whichever infectious disease so that you can control spread. And so in the top example we have here, we have without immunization, when you have the onset of infection, the infection passes to many individuals and people develop acquired immunity to the pathogen. And you have some that have acquired immunity, well, you can have potentially high mortalities in the population. On the bottom side, this is an example where you have an immunized population. So they're given a vaccine. Not everybody in the population may get the vaccine, but the majority of people get it. And so you've got, say, 80% of people who have some form of immunity against whatever infectious disease. And then this prevents a lot of the spread in the community. And as a result, you have a lot greater protection within the general population and a lot fewer fatalities to whichever disease it is. And so, you know, it's very helpful for this to have herd immunity, but really you need to have a high percentage of the population, depending on the infectious disease, in order for the population to control spread. For example, measles is a good example where you need to have about 95% of the population vaccinated for herd immunity. And we can see this now in isolated pockets in communities where there has not been as many individuals vaccinated that you're starting to see outbreaks of measles coming up. But one of the best examples of vaccine protection and eradication of disease is really smallpox. And we've seen that with other diseases that we have significantly reduced prevalence. So diphtheria, measles, and rubella when you do have consistent vaccination in the population. And so now to severe acute respiratory syndrome coronavirus 2, or SARS-CoV2 as it's called. So this is the causative agent in COVID-19 disease. It is a beta coronavirus. It's related to Middle East respiratory syndrome coronavirus, or MERS as it's known, and severe acute respiratory syndrome coronavirus, or SARS. It's a lot more similar to SARS than it is to MERS. They're both in the same family, but it's more around 80% similar to SARS and more like 35% at the amino acid level. Structurally, they're all quite similar. And in terms of the structure of the virion, the major surface glycoprotein is the spike protein. This is what decorates the surface of the virus. There's also, there's about a total of 29 total proteins. It's quite large RNA virus. And the majority of them are internal. There's also the envelope, membrane, and nucleocapsid proteins too that are quite important. And so in terms of cell entry, the spike protein on the surface is really important for cell entry. It binds to the angiotensin converting enzyme 2 receptor. This receptor is actually on a number of tissues. It's actually it's a lot less the distribution of the receptor is a lot less in the lungs compared to some of the other tissues, such as the kidneys, for example, but it is present in the lungs. It also is processed with by a serine protease called TMPRSS2, which will, I gather you'll probably hear more about it in the next e-seminar series with Stefan Pullman working, because his studies are really primarily on cell entry. So you'll hear a bit more about that next week. And what's really important about the spike protein is that it's immunogenic. It induces antibody and T cell responses, which are really important for antibodies, for vaccines, as well as for monoclonal antibody approaches. And really the majority of vaccines target this spike protein on the surface. So to give you an idea of the landscape of vaccine candidates that are being evaluated, there's over 150 different candidates in the pipeline. This ranges from preclinical candidates all the way up to those that are currently in clinical trials. And I think this is a lot more than we imagined at the start of this outbreak, that there would be so many candidates, but it's also a great thing because it shows all of the different approaches and strategies that we can use to develop protective immunity against infectious diseases. And so I've highlighted a few of the different partners who are working on various different vaccine candidates that are in clinical trials. And so there's other phase 1 trials that are anticipated to start this month and during the summer from many different companies and institutes, as well as multiple efficacy trials that are starting this summer. So if we now start to focus on gene delivery platforms for SARS-CoV2 vaccine delivery, we try to think about what we would want in an ideal candidate. So for example, an ideal candidate is something that is simple to administer. It delivers preventative or therapeutic control. It's readministrable, non-permanent, meaning that it does not integrate, and it can work in conjunction with already existing standard of care and supportive measures. And so I've divided this up into sort of 2 categories specifically for gene delivery platforms, viral vector delivery and non-viral delivery, such as nucleic acids. So if we look at viral vector delivery, these usually enter via receptor or cell target mediated entry, and are local injections primarily for vaccines, usually intramuscular. I've highlighted adenovirus vectors here, in particular human Ad5 vectors and a chimpanzee adenovirus vector. These 2 are currently in clinical trials. There are other candidates, such as measles vectors and others that are also being evaluated in preclinical studies. And so these are also other potential candidates for viral vector vaccine design. Some of the challenges though with viral vectors though is that many of these are single-use delivery. You can develop, a lot of people have pre-existing immunity against some of these viral vectors such as Ad5. For that reason, alternative vectors such as chimpanzee adenovirus vectors and others are being looked at because of the low seroprevalence in the human population. They do have long expression kinetics, which is nice in terms of inducing strong immune responses. On the other side, we have non-viral vector delivery. So we have both mRNA as well as synthetic DNA vectors. mRNA are typically encapsulated in lipid nanoparticles for delivery in a tissue, which can also allow for tissue-dependent targeting, depending on the LNP. For vaccines, it's usually local delivery. This enables localized delivery, but you can get some systemic expression. So even though the injection's at the muscles, sometimes you can also get mRNA dispersion into liver, lung, spleen, as well as other organs. It's nice because it's a transient expression. There's a short expression kinetics and it is readministrable. some of the challenges for mRNA are the dependence on the cold chain. So what this means is that mRNA requires refrigeration and storage, and they're working on different temperature stable formulations, but at the moment it requires like -80 storage. What we've seen due to the COVID-19 pandemic is that a lot of the processes for manufacturing and release have been dramatically accelerated. So before, this was quite a challenge with mRNA, but a lot of these have now been accelerated to try and improve and bring this faster so that we can accelerate this to the clinic. And then we have synthetic DNA vectors. So these are just plasmid DNA, local transfection of muscle or skin cells, and usually delivered using some kind of physical delivery method, such as adaptive electroporation. This delivers a transient vector expression and has quite a high consistency of transfection in the muscle, and it can be re-administered. There is no systemic expression, and it is cold chain stable, so you can also handle a large set of inserts. And so if we now focus a little bit more on DNA and RNA approaches, they really do have some theoretical advantages in terms of production timelines that can be favorable for rapid development of vaccines against infectious diseases. And so this is kind of the example that our group has followed, as well as the mRNA groups have followed, where you have the first case comes up, you get the sequence, and then immediately you start gene synthesis and rapid scale up. And for DNA, the cGMP DNA facilities are really well established. And RNA just takes a little bit longer because of the LNP formulations that are required, but those processes are also being accelerated. So I'll focus primarily the rest of my talk on synthetic DNA technology. I think what I really want to get across, I think, to this audience particularly, is that DNA delivery technologies have really advanced in recent years, and it's no longer the DNA that we've seen in the 90s and early 2000s. So I don't think anybody's really had an issue with DNA delivery other than that we've not seen great immunogenicity in people in the past. However, with these new delivery technologies, we're seeing dramatic improvements in immunogenicity as well as long-term induction of memory responses in people. And actually now these are quite comparable to what we are seeing with viral vectors. And so this DNA immunization is injected, you have local expression. This then attracts antigen presenting cells. It gets presented and you get priming of both CD4 and CD8 T cells, as well as B cells that start to generate immune responses. And a lot of the advancements in DNA technology have really happened because of gene optimization approaches. We have really high concentrated formulations now that are temperature stable. So we have over a year of room temperature stability. It's 4 to 5 years now at 4 degrees and at least a month at 40 degrees Celsius. And technologies such as adaptive electroporation have really enhanced local transfection of muscle or skin cells in order to really increase expression. And the other thing to highlight really is that plasmid DNA, at least specifically for gene therapy, has been in at least 15% of trials. There have been no serious adverse events. And it's really quite safe in terms of delivery to people. So to give you an idea of what this looks like then when we use adaptive electroporation, these are 2 examples with both intramuscular and intradermal delivery. So the DNA gets injected locally and then is followed by a short set of electrical impulses that basically cause transient pore opening to allow DNA uptake into the cells. And so on the left-hand side, these are rabbit muscle slices where you have no electroporation and with electroporation, so you can see a great enhancement of expression when you have electroporation. And on the right-hand side, this is intradermal delivery, showing that surface expression, cellular expression, and then within a few hours, you have infiltration of antigen presenting cells into the local site of delivery that then start to present antigen, and you get really nice increases in humoral as well as cell-mediated immune responses. And so the last two examples I'll give then for this, these are recent papers. So this one just came out recently in JCI Insight. This is an HVTN trial with delivery of a DNA-only vaccination using electroporation, demonstrating potent antibodies as well as CTLs against OMV using both intradermal and intramuscular formulations. And then on the right-hand side, with an intradermal DNA vaccine against Ebola virus, demonstrating that we can generate robust antibody responses. And actually these antibody responses are quite comparable to what is observed with some of the viral vector platforms, including the recombinant VSV vaccine. So now I'll focus on coronaviruses. And the reason I'm going to start with MERS is because this has really informed us for how to develop SARS-CoV2 vaccines, not just us, but other groups as well. We've learned a lot from the MERS vaccine developments and trying to apply this for rapid development against COVID-2. And so MERS is a very similar virus, and it has a subclinical infection to severe respiratory disease. It was first reported in Saudi Arabia, and then there was also a Korea outbreak in 2015. And the Korea outbreak was really one of, not the first time, but one of, in recent times, really highlighted the potential for global spread of infectious diseases, which we now see with the COVID-2 pandemic. And so when this MERS outbreak started, we designed a synthetic DNA vaccine. This encodes the surface spike protein. And this was, we delivered to different animal models, mice, camels, as well as rhesus macaques. This is the rhesus macaque data here, and showing that the vaccine could generate really robust T-cell responses and antibody responses. This particular study was done with intramuscular delivery. And we showed that this protected rhesus macaques against MERS coronavirus infection. So both a high dose of the vaccine as well as a low dose of the vaccine protected against MERS-associated pneumonia, as well as viral loads in the lungs. And so based on this promising study, then we then went into a phase 1 trial. This was in collaboration with Gene1 Life Sciences and the Walter Reed Institute of Army Institute of Research. The clinical PI on this was Kayvon Majard. And so these are antibody titers in people. So following intramuscular administration, we observed durable anti-spike IgG responses. These responses, even at the lowest dose of DNA vaccine, lasted for at least a year. We don't have time points yet for later time points, but this is at least a year. And on the right-hand side, this is comparing to what we see in terms of antibody titers between acute illness, convalescent MERS patients, as well as the vaccine. And so the vaccine is quite comparable to what's observed in MERS convalescent patients. And this is important because at least from what we see, we believe at the moment that previous MERS infection is protective against subsequent MERS infection. And so in addition to antibodies, we also looked at T-cell responses. And across all of the dose groups, even at the lowest dose, we're seeing robust T-cell responses against all regions of the spike protein. And actually these T-cell responses are significantly higher than what we observe with the MERS convalescent patients. We also have T-cell responses for week 60 that are higher than the convalescent patients as well. That's just not in this paper, but we do have that. And so based on this data with intramuscular group, with the intramuscular delivery, we've also been evaluating intradermal delivery. So intradermal delivery, the reasons for that is that it can be potentially dose sparing, and also it's more tolerable route of delivery as an alternative to intramuscular administration. And so we've recently been evaluating the MERS DNA vaccine using intradermal delivery. And so we delivered a low dose of MERS and demonstrating that we can induce robust antibody responses that are comparable to intramuscular delivery. The T-cell responses do trend to be a little bit lower. but we're generating robust T cell responses. And then in challenge, in MERS challenge, we have protection compared to the control groups against this MERS installation challenge. The other important thing to highlight is that we do not have any enhancement of infection in the vaccinated animals. And so this is, these studies have really laid the foundation for our SARS-CoV-2 vaccine design. And so really what I wanted to highlight with the MERS section is that ID-delivered vaccines can elicit strong immune responses, and that ID delivery of DNA is dose sparing even at the lowest doses. And this really supports delivery using this highly tolerable route. And these studies really lay the foundation for accelerated DNA vaccine development against COVID-2 in pretty much all of the aspects. So design, delivery, formulation, dosing, as well as regulatory filing. And so we have been working on MERS all of this time, and then the COVID-19 pandemic started. And so we rapidly shifted over to designing a DNA vaccine against SARS-CoV-2. So on January 11th, 2020, the virus sequence became available on GSAID.org. And so we looked at the sequences. At that time, there were only 4 sequences, and now there's 34,000 sequences plus and growing every day. But at that time, there were only 4 sequences available. And so we immediately started to analyze the sequences, look at them and see how they were structurally compared to SARS, the original SARS-CoV-1, as well as MERS-CoV, which we have a lot of experience with. And structurally, they're quite similar. And so this really helped us with our gene optimization. And our group settled on working with a full-length SARS-CoV-2 spike antigen. This was cloned into our plasmid DNA vector and then expressed to confirm that we had expression of the spike protein. The next thing that we did is then we went into animal models. So we delivered a single dose, single immunization in mice as well as guinea pigs to look at humoral responses as well as T cell responses following delivery of the COVID-2 vaccine, which is INO 4800. And we observed really rapid induction of T cell responses. 7 days after administration, we're starting to see T cell responses, which continue to increase. We also have robust binding of sera from immunized animals to the receptor binding domain protein. This receptor binding domain, these are blocking antibodies. We also show in this paper that these antibodies from mouse sera can block that interaction between spike and the ACE2 receptor. And so we're also seeing that even at our lower doses, we're getting very robust endpoint titers against the RBD. In the paper, we also show that we have binding against other parts of the spike protein as well. And similarly, we see the same thing in guinea pigs, which is at the bottom. We further go on to show that in the lungs of animals, so when we do bronchoalveolar lavage in mice as well as guinea pigs, we have detectable anti-spike IgG in the lungs. So this is the site of infection. And so this is also very promising to show that we can get antibodies for a vaccine that's delivered in the muscle or intradermally going into the lungs. And we also did T cell mapping in mice to show that we're generating T cell responses, not just against one part of the spike, but against multiple regions across the spike protein. In bulb C mice and now in Black 6, we've identified several immunodominant epitopes. And some of these epitopes are quite similar to what's been observed in the past for human epitopes for SARS-CoV-1. So there's a lot of, the vaccine is able to generate significant breadth in terms of both antibodies as well as T cell responses. And so more recently now, we've moved into other animal models such as rabbits and non-human primates. So this is data from showing our T cell responses and antibody responses following immunization in rhesus macaques. This is a 2 immunization regimen delivered intradermally. And we show that we're even at week 2, we're starting to see development of robust T cell responses. And following the second immunization, we have really strong T cell responses. The same with antibody responses. And importantly, we have 100% seroconversion after a single dose. The antibody responses that we're seeing are actually quite similar to what's being observed for convalescent sera that's coming out in the clinic. So this is really interesting and supporting the immunogenicity of our SARS-CoV2 vaccine. And so I just wanted to end then on the clinical trial now. So all of the work that I showed you with CoV2, that was pretty much accelerated in less than 3, I'm kind of trying to calculate, yeah, just in 10 weeks. So we quickly did all of the animal work. The mouse and guinea pig work went towards the FDA filing, and we were allowed to begin on April 2nd. So this trial is currently ongoing, evaluating safety, tolerability, and immunogenicity. And so hopefully we'll start to hear soon about immunogenicity coming from this phase 1 trial. Also, our partners at Inovio have now announced that they're going to advance this into a phase 1/2 trial in Korea. And we're looking forward to seeing advances towards an efficacy program this summer. So all of this has been the result of a lot of hard work from many, many people all over. Our funding comes from CEPI, which is the Coalition for Epidemic Preparedness Innovations. This has really been a team effort from Inovio, the University of Pennsylvania, as well as the Wistar Institute, as well as many different funding agencies and collaborators, really trying to accelerate a lot of the COVID-2 vaccine development and moving this quickly into people.
Thank you very much.
Yeah.
Thank you very much, Amy. This was very interesting, and we were very clear. So it was a great talk.
Thank you.
There are some, I can see one question from the audience, which is actually not really related to vaccines for COVID, but it's more general. The question is, can COVID-19 spread through food?
To my knowledge, I have not heard any reports of it being spread through food. I feel that it's a lot less likely. You're more likely to get it through person-to-person spread.
Right. So until we get some more questions from the audience, can I actually ask you some question? So in general, sure. in general, when you use plasmid DNA as vaccine, as in your case, what is the longest time point at which you were able to detect a persistent protection? Or, I mean, you or others? I mean, it's more of a general question.
Yeah, so I mean, in terms of protection in animal models, some of our colleagues have done studies where you immunize the animals and then give them like a viral challenge after a year, and you still have protection, for example. Okay. So we've seen that, yes.
Okay, and if you were to go longer than a year, do you think you needed to boost again with the vaccine, or with?
I think it would depend on the infectious disease. So I don't think anybody's done longer than a year that I know of, just because of the cost of keeping the animals. That's just the only reason that nobody's done it. But I think it depends on the infectious disease and what kind of boost you would see. So we've done studies with the Ebola vaccine in monkeys where we did not challenge after a year, but we gave them a boost after 1 year, and you see a dramatic recall response immediately after you give the boost. So it could be the same way. I think it just depends on the infectious disease.
Okay. There is another question from the audience. Which by the way starts with excellent talk, and then it says, what is your outlook regarding adverse events occurrence with your new DNA vaccine in COVID-19?
Yeah, so I think it's always difficult to predict what adverse events may happen, but historically what we've seen with DNA vaccines is that we have not seen any serious adverse events. Any events have been just from local injection site pain that quickly dissipates. We have not, with the MERS coronavirus DNA vaccine, we've not had any serious events. It's very similar coronavirus. So I think that is really positive for the COVID-2 DNA vaccine.
Okay. Another question from the audience, which vector vaccine approach do you think will be available first?
That I think is also really hard to tell. So there's a lot of different groups that are testing out their vaccines right now. And I think it's just, everything's being accelerated at the same time. So we've, AstraZeneca and Oxford are now moving the ChAdOx platform, encoding the COVID spike really rapidly into efficacy trials. Moderna's really rapidly advancing their mRNA. DNA will hopefully start into efficacy. So it's everybody's kind of, I guess, neck and neck. But I don't want to say that we only are going to have one vaccine candidate. I think we're going to need all of these multiple different vaccine candidates, whether it's viral vector or non-viral vectors, just given that we need to cover, 7+ billion people. So we're going to need many different vaccine candidates, particularly because some vaccine candidates may be better for certain populations, such as immunocompromised individuals, the elderly, pregnant individuals, as well as others, such as diabetics.
And another question, I'm not sure I actually understand it, but it's antibody-dependent enhancement in coronaviruses.
Yes, so I guess they're asking about whether we're concerned about antibody-dependent enhancement. I think that's a question we've had a lot. There's this, I like to call it a theoretical concern. So there's a theoretical concern that giving vaccines or an antibody could enhance disease through different mechanisms. We see that with dengue virus. To date, we have not actually seen that in people with coronaviruses. A good example is like the MERS vaccines, and also COVID so far, there hasn't been any enhancement of disease. We've seen this in animal models, primarily mouse models, and with certain viral vector candidates. Whether or not this is a real phenomenon in people, we're still not sure about that for coronaviruses. And I guess one of the supports for not having antibody-dependent enhancement so that there is no disease enhancement is really that there's been thousands of individuals that have received convalescent transfer of sera that have basically no neutralizing antibodies or very little antibody levels, and we don't see any enhancement of disease with convalescent transfer of plasma. So at the moment, I mean, it's a concern in everybody's mind, and we're of course testing all of the possibilities, but it still remains a theoretical concern.
Theoretical concern, I see.
Yeah. And
then there is another question about the possibility to predict virus drift mutation. In other words, if virus evolves over time, and then can we adapt our vaccine to this evolution?
Yeah, that's something that us as well as other people in the field are trying to track and to understand now that we have tens of thousands of genomes sequenced for COVID-2, trying to see how the virus is changing. Compared to other RNA viruses, SARS-CoV2 is not changing that rapidly. Part of it is because it has this internal proofreading mechanism, which is unique compared to some of the other RNA viruses. But we are trying to follow it to understand which regions of the virus, whether it's internal proteins or the external spike protein, are changing over time. It seems primarily to date, there have not been many changes in the receptor binding domain, So that actually works in favor for the majority of vaccine candidates that are being evaluated right now, because those are really trying to target that spike ACE2 binding interaction to prevent that from happening.
And actually, because you've been working also with the DNA-encoded antibodies, do you think this can be an approach also for SARS-CoV2, or? Yeah,
definitely. And we're working on some of those different approaches. The advantages for DNA delivery of antibodies is that you can get very long-term expression of antibodies. So recombinant antibodies have some benefits for therapeutic delivery, but we can now also consider delivering antibodies as preventative measures, so that you can have antibodies delivered if someone's going to be particularly at risk, or also in conjunction with vaccine, because vaccines do take some time to develop protective immunity. So you could think of those as more of a preventative approach for delivering antibodies using DNA, in addition to the recombinant antibody biologics for therapeutic delivery.
There are additional questions. One is what do you think about the RNA vaccine approach? I guess the Moderna approach. Do you think it is a promising approach?
Yeah, I think the majority of approaches that are being tested, mRNA, viral vectors, DNA, are all quite promising. They all induce immunity slightly differently. mRNA from previous studies can induce antibodies and CD4 T cell responses. We're still waiting to hear how their CD8 T cell responses look. At least for other coronaviruses, CD8 T cell responses for MERS have been associated with better outcome following infection. But I think it's quite promising. They've definitely accelerated development quite quickly, and so we're looking forward to hearing how a lot of these different platforms are, how well they will turn out in efficacy trials.
Sure. Somebody else is asking, what is the plasma therapy?
Oh, so the convalescent plasma therapy. So that's actually an old, it's been around for quite a, I mean, I want to say like 100 years. And so it's basically transferring plasma from convalescent patients, so people who've recovered from an infectious disease, and transferring their plasma to other individuals to help protect them or help treat them against the same infectious disease. Mm-hmm. So basically, it's a passive transfer of antibodies for protection.
Right. And then how do you see manufacturing of your DNA vaccine if this gets approved? How challenging is going to be to, I don't know, produce 4 milligrams per individual of plasmid DNA? How many people do you think can be treated?
Yeah, so I mean, cGMP plasmid production is actually quite well established. And there's facilities across the world that are capable of cGMP production of plasmid. It's actually usually the first step in a lot of other vector production approaches. And so in terms of capacity, I mean, there's always a need for increased capacity for facilities to produce more plasmid, and just, or for any product really. But I think it is feasible. And that's one of the reasons, particularly in the MERS study, that we started looking at lower dose delivery as well as intradermal delivery, because that further reduces the amount of DNA that we need to give in order to see the same levels of protection. So we're working on strategies really also to lower the amount of DNA that's required.
Okay. And as a curiosity, are there other SARS-CoV2 antigen that you think would be amenable for a vaccine other than spike?
Yeah, so I think, I mean, the main reason where we and others have chosen spike is because it's the surface glycoprotein, but there are others I think that may be useful in conjunction with spike. So some of the internal proteins may be good at generating T cell responses. And so those might be nice to pair with spike so that you have a really robust antibody responses, but you're also getting good CD8 T cell responses that are being generated against many of these internal proteins.
Okay. And what is the anticipated date for approval if everything goes well and efficacy is proven?
I mean, I think that's hard to predict right now. I think we need to go through some of these efficacy trials. Us and everybody else, we're trying to accelerate vaccine development, but we still need to make sure that we're going through all of the clinical trials appropriately and assessing, immunogenicity, efficacy, and everything appropriately. So it'll still take some time. It's not as quick, unfortunately, as we would like it to be.
I'm sure, yeah. Okay, I don't see other questions from the audience. So thank you very much for your talk and for being so exhaustive in your answers and comprehensive. Thank you very much.
Thank you very much.
Bye-bye.
Bye.